All right, so we've talked about how infectious microorganisms, pathogens, have to enter the body and then they have to attach to cells and or tissues in the body so that they can stay in place. And a third critical step for organisms that cause infectious disease is they have to be able to survive our host defenses. So when we get into chapter 12, we're going to talk about these first and second lines of defense of the immune system. So barriers and weapons. We've already talked about barriers, your skin and your mucous membranes, but we'll talk more about the features of those barriers that help prevent entry of microorganisms into the body. But then we'll also talk about the second lines of defense when we get into chapter 2, which are some of these early weapons, both cells, and then there are molecules that function as early weapons against invading microorganisms in the body. So it's these early weapons that pathogens really need to be able to get around. So different pathogens have different strategies for accomplishing this, and I'm actually going to expand on the textbook information, so be sure you're aware there will be some material that I'm covering here that I'm really pulling out from later parts of the textbook and bundling in here with chapter 11. So pathogens have ways of either avoiding, evading, or compromising host defenses, or doing some combination of all three. And so some of these methods can be considered passive, and those are just structural features. Some of those we've talked about before, like bacteria that have capsules. Those capsules make it more difficult for white blood cells to do phagocytosis and engulf them. And then others are active methods. So these are really weapons that pathogens produce in some cases that are able to harm our immune system. So they actually prevent the immune system from attacking them by going ahead and attacking the immune system. So pathogens have weapons they use against our immune system, and then our immune system has a number of weapons that it uses against pathogens. So your non-pathogenic microorganisms simply can't get around. They don't have these capabilities. They have no way of getting around those early forces of your immune system, and so once they enter the body, they tend to get wiped out, and that's why they're never able to stick around long enough to cause disease, as long as you have a normal, healthy, functioning immune system. Now this differs in patients who are what we call immunocompromised. If you have a weak immune system, then all bets are off. There are a number of microbes that don't cause disease in healthy individuals that do cause disease in patients that have a weak immune system. And we'll learn more about that as we get into the last unit for the course. So what are some passive methods for evading host defenses, for evading the immune system? All right, so capsules. So capsules occur on bacteria. Make sure you make a note of that. You don't have capsules on viruses or protozoans, things like that. And if you guys remember, a capsule is a very thick, organized glycocalyx. A glycocalyx is that outermost layer that occurs on some bacteria. Not all bacteria have these. A number of pathogens have capsules, though, and I believe your textbook has a listing of some organisms that do have capsules. This is a picture of a capsule stain. You guys did that as a virtual laboratory exercise, and that's a special type of staining that stains the main cell part of bacteria and also stains the background. Remember, that was called a negative stain. And then you have a clear zone here, and that's where the capsule is located. The capsule is not taking up any of the dyes that were used. So you can see that those capsules can extend pretty far off of the bacteria. And the reason that this helps pathogenic bacteria that have capsules escape infection is because the cells, your white blood cells that do phagocytosis, can't latch on to these bacteria as well when they have a capsule. Now, interestingly, this is a temporary advantage. Your immune system has early defenses and it has later defenses that are much more powerful and specific about what they're attacking. And so later during an infection, your immune system will actually generate antibodies. Antibodies are weapons that attach to very specific things and tag them for destruction. And so you'll actually make antibodies that attach to the capsule of a pathogenic type of organism. You just have to make sure you live long enough to make that happen. So there are pathogens that have capsules that are capable of killing many of their hosts if you don't get treated for it. So a good example of a bacterial organism that has a capsule is Streptococcus pneumoniae. Now there are strains of Streptococcus pneumoniae. Now we talked about this back in the genetics chapter. This was a discovery that led to an understanding of genetic transformation in bacteria, if you guys remember. The Griffith experiments that we talked about that were done in mice. But Streptococcus pneumoniae is a bacterial organism, causes many cases of bacterial pneumonia, but there are strains of it that don't make capsules, so they lack the enzymes that create the capsule layer, and those strains are not capable of causing disease. Now a little bit of a reminder about phagocytosis, and we'll be talking more about this in chapter 12 as well. This refers to engulfment of microbes or damaged or dead cells from your own body, damaged tissues, etc. So there are white blood cells and their relatives whose job it is to do phagocytosis. They're kind of like the vacuum cleaners of the body. And when they do this, here we've got a poor little bacterium that's going to be phagocytosed by this white blood cell over here. And so the white blood cell attaches to the bacterium, engulfs it, brings it inside in a membrane-covered compartment. This is called a vesicle or a food vesicle. And then that compartment merges with another little membrane-covered compartment called a lysosome. So a lysosome is one of those eukaryotic organelles that back in chapter 4 I mentioned we weren't going to go over the laundry list of eukaryotic organelles. I would just bring them up on an as-needed basis later in the semester. So now it's later in the semester. So lysosomes are membrane-covered compartments in our cells that are full of digestive enzymes. And so phagocytic cells are going to have a lot of those. A lysosome will merge with the food vesicle over here that has the little microbe in it, dumps all these digestive enzymes in here, and those digestive enzymes murder the poor little microbe, break it up into subunit molecules, break apart all those organic molecules that make up the microbe. And then that vesicle, which is now called a phagolysosome, travels back to the surface of the cell and spits out all of those waste contents through a process called exocytosis. You're eliminating or secreting those products. All right, so when we get into chapter 12, we're going to talk more about your professional phagocytes, and macrophages and neutrophils are going to be two types of cells in the body that are known to be professionals at doing this. So bacteria that have capsules are good at avoiding this step right here. These macrophages and neutrophils can't latch on to encapsulated bacteria very easily. All right, so there are a couple of other good examples of pathogenic bacteria that have surface features that prevent phagocytosis. And and this is something that's not covered in your text until later in the textbook. It's not going to be in chapter 11. But a good example, so this is an electron microscopic image of Streptococcus pyogenes. That's the organism that causes strep throat, among other things. And Streptococcus pyogenes does not have a true capsule, but it is coated across its surface with little protein spikes that are called M proteins. So these little things that kind of look like hairs sticking out of the bacteria. Now those aren't cilia like we were talking about that line your trachea. You know, here we're talking about a bacterial feature, much smaller, and they're simply little protein spikes called M proteins. But they kind of function like a capsule in that they prevent neutrophils and macrophages from being able to latch on to the bacteria easily. Now these things do stick out of the surface. Later during infection, your immune system will actually make antibody weapons that attach to those M proteins. So these little spikes are useful early during infection, but not so much later. Now over here we've got an electron microscopic image of Mycobacterium tuberculosis, which causes TB. And we've tore about how it has a special cell wall made from mycolic acids. Those are a waxy form of lipid. And those also inhibit phagocytosis. So your macrophages and your neutrophils can't latch on to Mycobacterium tuberculosis quite as easily as they can other types of microorganisms. Those mycolic acids also protect mycobacteria from drying out, desiccation, various disinfectants, antiseptics, antibiotic drugs. We mentioned that back in chapter 10. The mycobacteria, we don't have nearly as many drugs that can penetrate that mycolic acid cell wall and get in there and do harmful, nasty things to the bacteria. All right, so you want to keep in mind some of these examples we've talked about of surface features, and these have all been bacteria that are these examples that we've just gone over. So not viruses or protozoans or helminths. Things that help inhibit phagocytosis. Now those other kinds of microbes will have other features that may help prevent phagocytosis, but the examples that we've gone over here have all been different kinds of bacteria. All right, so those are passive examples of passive methods of avoiding the immune system. What are some active defenses? So these are going to be actual weapons that these microorganisms secrete to try to harm your immune system. And there are a number of examples of these. Leukocidins are produced by some Staphylococcus and Streptococcus pathogens. So those are genus names of bacteria. And these are proteins, and what they do is they will embed themselves in the membranes of our white blood cells, and they create protein channels there. So they're basically hole punchers. They're poking holes in the membranes of the white blood cells, and that causes those white blood cells to die. So that's a pretty nasty kind of weapon. Incidentally, this hole punching I'm talking about, we're gonna see a number of examples of that, because there are pathogens that produce proteins that attach to our cell membranes and create pores in them and cause our cells to die. And then our immune system secretes various kinds of proteins that do that to microorganisms. So they have weapons that do that, and we have weapons that do that to them as well. All right, then hemolycins also more or less do the same thing. These are proteins that attach to cytoplasmic membranes of our cells and poke holes in them and cause those cells to lyse or burst. And so these tend to cause red blood cells. Hemo refers to the blood. So hemolycins will cause red blood cells to burst and then white blood cells as well. So again, the Staphylococcus genus, the Streptococcus genus have example species that do this. And then good old Clostridium perfringens, if you guys remember that, that's the cause of gas gangrene. That organism produces hemolycins as well. So that's kind of nasty stuff. Now a couple of enzymes that are involved in helping defeat the early forces of our immune system. Staphylococcus aureus produces an enzyme called coagulase. So remember, ASE is generally referring to an enzyme. And what this enzyme does, hopefully that's reminding you, if you've had Biology 202, of blood coagulation, blood clotting. And what this enzyme does, it's a bacterial enzyme. So these are supposed to be some bacteria over here. And they're secreting or releasing this protein enzyme called coagulase. And what that coagulase does is it triggers the formation of a clot using fibrin protein. So you guys learn about blood clotting when you take Biology 202. So basically the bacteria are hijacking the fibrin that's in your blood and is supposed to help you clot blood when you need to surround themselves in this clot, and then that's a method for them to hide from the immune system. So that's pretty sneaky. If you've ever had a boil before, that's kind of like a pimple from hell. Boils include these coagulated masses with the bacteria on the inside of them, just in case you wanted to be grossed out a little bit more. But while the bacteria are inside that clot, they're hiding from the immune system. And then those bacteria that make coagulase tend to also produce kinases, and kinases help dissolve clots. So if the bacteria they'll encase themselves deliberately in a clot so they can grow inside there and hide from the immune system. But at some point you got to escape and move on to a new host. You've got to be transmitted elsewhere. So kinases are enzymes that will help dissolve clots. Also clots form naturally to try to help seal off wounds and so forth. So these kinases that dissolve clots can help these organisms escape from that as well. Staphylokinase and streptokinase are examples of enzymes that dissolve fibrin clots in the body. And interestingly, those are being used now deliberately as drugs to help dissolve clots in heart attack and stroke victims. I did want to point this out just kind of as a little sidebar. So over here we mentioned that there are some organisms that produce hemolysins. So those are proteins that will cause red blood cells to burst, and also some white blood cells as well. So that feature can actually be used to help us identify certain types of bacteria. So long ago, microbiologists noticed that when they spread bacteria onto agar plates that contain 5% sheep's blood, so you can actually make these in the laboratory, it's not too hard, there were some types of bacteria around which you would see a clear zone. And it turns out that that clear zone, like you see right here, is because those bacteria are secreting hemolysins that cause red blood cells to burst. And so you'll see a clear area surrounding those bacteria. So if that happens, that's called beta hemolysis on the plate. So if that occurs with a species of Streptococcus bacteria, that's called beta hemolytic strep, and that can be used to help you narrow down the identities of members of the Streptococcus genus, of which there are a number of different pathogens. All right, so if you have bacteria on a blood agar plate, and if you guys are around clinical microbiology labs, you're going to see lots of these blood agar plates. They're very commonly used. The blood has a lot of growth factors in it that many pathogenic bacteria like. So many microbiology labs and clinical facilities will just kind of routinely use these blood agar plates to make sure that whatever they spread on there is likely to grow pretty well. So that's the Greek letter beta, by the way. So this bacteria that are beta hemolytic were spread on this plate deliberately in that pattern. And over here we have the Greek letter alpha, and you can probably see there's a green zone around there. And that's not really the red blood cells bursting, but instead there are substances released by the bacteria that cause the iron atoms in the hemoglobin to become oxidized. So they lose electrons and they take on a green color. So that's not really true hemolysis. That's not where the cells are really bursting. It's just a change in color of the hemoglobin due to changes in the iron atoms, oxidation. That's called alpha hemolysis. And finally you have gamma hemolysis, like the bacteria here, which really means no hemolysis. I don't know why the terminology is so confusing, because the only real hemolysis here is beta. The other two are not true hemolysis. And I especially don't understand gamma hemolysis, which means no hemolysis. I can't, that's just the way where the terminology, it's older terminology, and it's still used today. So you'll sometimes hear this terminology being used if you're around a clinical microbiology laboratory. So just to show you how this can be used, so if you have an organism that you know is a type of Streptococcus or one of its close relatives, and you can tell that by looking under the microscope and observing the cell morphology. The beta hemolytic species of Streptococcus include Streptococcus pyogenes and Streptococcus agalactiae, or agalactiae. So they're both beta hemolytic, and Strep pyogenes is the cause of strep throat and some other types of infections as well. It actually causes some cases of flesh-eating disease. And then you have your alpha hemolytic strep, so they both have a green zone on the blood agar plates, and that includes Streptococcus pneumoniae, and then a group known as the viridans Streptococcus, which includes two species, Streptococcus mutans and Streptococcus sanguis. And then finally, gamma hemolytic, the Enterococcus genus, is closely related to the Streptococci and includes Enterococcus faecalis and Enterococcus faecium, and these sometimes cause infections. They naturally live in the intestinal tract, but sometimes they get into other parts of the body like the urinary tract and can cause infections there. So anyway, you can kind of see how the pattern on these blood agar plates can help you start to at least partially identify these organisms. And this term, and to make matters worse with this, among your beta hemolytic streptococci. Streptococcus pyogenes is sometimes also called group A strep, and Streptococcus agalactiae is sometimes also called group B strep. And group B strep can be a problem, especially in newborns and people with low immunities. It can sometimes cause meningitis or sepsis. Sometimes there's concern with newborns. If the mother is found to have Streptococcus agalactiae living along her vaginal lining, they have to take precautions with the newborn, right after birth to make sure they don't develop an infection with those bacteria. Your group A strep over here, Streptococcus pyogenes, that's the organism responsible for good old-fashioned strep throat. But a lot of this can get kind of confusing, because you'll hear people talking about group A strep and group B strep, but both of that has nothing to do. A is not alpha, B is not beta. So both of those fall into the beta hemolytic category. So I just wanted to point that out. You've been exposed to it in case you see it down the road. Some other active defenses. The first line of defense in your immune system, as we'll learn when we get into chapter 12, are the skin and the mucous membrane barriers. So some microorganisms secrete enzymes that break down those barriers. So a couple of examples are high hyaluronidase, I know that's kind of a hard word to say, and collagenase. High hyaluronidase is an enzyme that breaks apart high hyaluronic acid. That's a type of carbohydrate that's kind of like a tissue glue. It helps hold cells together. And then collagenase breaks down collagen. So when you guys take Biology 201, you learn about how your connective tissues, the spaces in between cells, the extracellular matrix, has a lot of collagen proteins in it. So if you can break down the collagen, that's going to help you invade deeper into the body. So what they're showing you up here, you've got some bacteria that secrete high hyaluronidase enzymes, and that's helping to break apart the connections between epithelial cells. Remember, the epithelial linings of the skin and the mucous membranes, those cells are tightly packed together, and that's what helps give them barrier properties. So if you produce enzymes that break those connections, that's going to help you invade. And then once you get down in here deeper, there's a lot of collagen fibers around, so if you can break those down, that's going to help you invade as well. Plus the bacteria, you break down collagen. Collagen is a protein, high hyaluronidase is a carbohydrate. If you can digest those and break those down into building blocks, the bacteria are likely to be able to use those building blocks for their own purposes, including energy. So it's a type of feeding as well that's taking place. Some organisms that make high hyaluronidase and collagenase include Clostridium perfringens, which causes gangrene, Streptococcus pyogenes. There are some strains of this organism that cause flesh-eating disease, which is also known as necrotizing fasciitis. Necrotizing refers to necrosis, which is tissue and cell death. Fasciitis is inflammation of the fascia. So if you guys remember from Biology 201, fascia, you have some connective tissues in between the skin and the muscles and the bones. And so if you have flesh-eating disease, you have inflammation within that area taking place. And I know that's a gross, nasty picture. If you want to really gross yourself out, Google necrotizing fasciitis. You'll find all kinds of nasty pictures where you see tissues with holes in them and liquefaction of tissues. Pretty nasty, gross stuff. All right, another active defense that some bacterial organisms are capable of is hiding inside cells. And this is kind of weird. So we just talked about phagocytosis. There are some types of bacteria that allow themselves to be phagocytosed, and then they produce substances that prevent the lysosome from merging with the food vesicle. So they just stay inside this nice little membrane-covered compartment inside a phagocytic cell, like a macrophage or a neutrophil, and they increase their numbers. They reproduce in there. They never get digested by all those digestive enzymes in the lysosome. And then interesting, Listeria monocytogenes, or listeria, causes listeriosis or listeriosis. And they have kind of an interesting strategy. They will allow themselves to be phagocytosed, but then they have a way of breaking out of the phagolysosome before they get destroyed, and they will proliferate, so they'll multiply intracellularly inside one of your cells. And then what they do is they actually hijack the actin in your cells. So actin fibers form parts of the cytoskeleton in our cells, and then you guys also learn a lot about actin when you study the muscular system in Biology 201. That's a protein fiber that's really, really important for muscle contraction. So Listeria bacteria have evolved a way to hijack actin, and they use the actin to propel themselves from one cell to another. They use the actin to more or less kind of push them from one cell into another, and that way they just spread from cell to cell and multiply in cellular hosts, and they're escaping from the immune system. Your immune system weaponry is all out here, and by staying inside your cells, they're actually hiding. So that's kind of a nasty little strategy that the Listeria bacteria have developed. All right, so that concludes some example methods by which pathogens, and again, we mainly focused on bacteria in that particular video lecture. In the next one, we're now going to talk about, well, how do these microbes damage us? Now, we just saw some examples of damage, but we'll talk about even more examples of damage as we get into this next video clip and talk about step 4, actually microorganisms causing disease, damaging us, causing signs and symptoms. You know you're sick, you know you're feeling bad. How is all of that happening? And in video lecture 6, we'll talk about more enzymes that can cause that, and also something called bacterial exotoxins. We'll talk about those as well.